This talk is entitled, Using a Knowledge Base to Facilitate Post-Translational Modification Assignments.
Written by Ronald Beavis, Daniel Evans, and David Fenio.
By way of introduction, Tandem is an application for modeling peptide sequences, based on putative peptide tandem mass spectra for the purposes of protein identification and the discovery of post-translational modifications. The capabilities of Tandem for detecting post-translational modifications have been altered by the development of a knowledge-based approach, which uses existing protein annotations to facilitate the detection of modifications that are not common in the proteome in general, but frequently found in specific proteins. The approach used was straightforward to implement and required minor changes in the Tandem codebase. The approach is generic and can be implemented in any similar peptide identification algorithm.
The tandem mass spectra obtained for the identification of peptides and proteins in proteomics are compared to existing protein sequences using algorithms based on several different approaches. Currently, the most common approach is based on Lederberg and others, 1969, as applied to peptides by Eng and others, 1994. Peptide sequences are generated from protein sequences and compared with spectra using general rules associated with the sequence-specific fragmentation of peptides, Beeman and Martin, 1987. Tab and others, 2007 discussed the various algorithms that utilize this approach.
This approach has been popular for proteomics data analysis, mainly because it does not require the prior observation of a peptide to identify it. All the analyst needs to specify are some instrumental tolerance parameters, and which post-translational modifications, PTMs, are to be considered during the analysis of an individual dataset. The number of PTMs selected will normally be minimal, as the time required to perform the comparison depends on the number of potentially modified residues in that sequence. If there are n modification sites and each site has only one modification allowed, then the time required to analyze that sequence scales as n to the power 2. This scaling factor causes serious slowdowns in calculations, particularly in protein sequences with low complexity regions that result in large n values for multiple peptides. In practice, investigators often analyze their data for only the most common modifications caused by experimental artifacts, for instance, metoxidation, rather than the less common, but potentially more interesting PTMs.
The rest of this talk describes our implementation of a knowledge-based approach to reducing the number of calculations necessary to check routine datasets for proteins with known PTMs, in addition to any modifications the user might wish to test for in the entire dataset. A knowledge base was initially constructed using information made available by Uniprot, Bolkman and others, 2005. NZMBL protein accession numbers were mapped to Uniprot accession numbers using the NZMBL Biomart, Smedley and others, 2009. The Uniprot was then parsed to create a list of known PTMs correlated with Uniprot accession numbers and mapped back to NZMBL accession numbers. These modifications were then translated into the BioML, Fenio, 1999, format used to pass commands into Tandem, and written into an XML file that served as our knowledge base.
For the NZMBL version 56 of the human proteome, this process resulted in commands for 10,525 protein accession numbers. The specific modifications selected from Uniprot were as follows: serine, threonine, tyrosine and histidine phosphorylation, aspartic and glutamic acid carboxylation, aspartic acid, lysine and praline hydroxylation, tyrosine sulfonylation, tyrosine thyroxylation, arginine and lysine methylation, lysine acetylation, ubiquitination, and phosphopyridoxylation. Many NZMBL protein sequences may correspond to one Uniprot entry because of alternate splicing and other differences between the two sequence collections.
An alternative approach would be specifying the exact residue that should be tested for a PTM, rather than testing all appropriate residues in a given protein. This method was tried, but two problems were encountered applying it to real data. Firstly, it was difficult to make exact one-for-one residue maps between a basis set of sequences, for instance, Uniprot, and another sequence set. Secondly, the variability of the biological formation of PTMs can make it hard to know in advance exactly which residues may be modified. Both of these problems resulted in reductions in the number of high-quality identifications obtained using this alternative approach compared with the coarser, but more inclusive, protein-by-protein method.
To take advantage of the knowledge base, two changes were made to the basic algorithm that runs Tandem. The first change was the addition of a method that loads the knowledge bases necessary for a given set of protein sequence FASTA files by constructing a C++ standard template library map object using the protein accession number as a key and the modification specification as data. This method uses the same configuration file used to generate the list of FASTA files to be used for a particular species, with added specifications for the modification knowledge base files.
The second change was to the portion of the algorithm that loads an individual protein sequence. A step was added to utilize information loaded in from the knowledge base. After a protein sequence was loaded, its accession number was used to look up the PTMs in the map object just mentioned. The PTMs were then checked so as not to conflict with PTMs specified by the user at runtime and the processed list of PTMs for that protein was stored for use at runtime.
In practice, the use of this new method of testing for PTMs was more than 20 times faster than testing for the PTMs using conventional methods. It also resulted in the discovery of phosphopeptides and peptides from samples containing collagen, histones or ubiquitin that would not be found using standard proteomics data analysis methods.
In conclusion, the use of a knowledge base to specify PTMs for specific protein sequences, rather than testing all protein sequences for all PTMs, has the potential to facilitate the more thorough analysis of proteomics data. The knowledge base approach taken was not algorithm specific. Most other peptide identification algorithms could be modified to use the same scheme and obtain similar results.